hopefully you found that it says, I love learning about genetics. So that is an example of a code, that you've probably seen sometime in your life. However, the genetic code is one that the cell uses, and it tells the cell how to read the DNA message. And it reads the DNA message and uses that DNA to make a protein. So the genetic code tells that cell how to take that DNA message, which is written in those A's, C's, T's, and G's, and turns them into a protein. There's 2 different steps to this process. The first step is called transcription. So transcribe, right? Transcription is basically to rewrite. So you're going to take DNA and you're going to rewrite it in a form called RNA. I'll show you what RNA looks like in a moment. Then you're going to translate the DNA, and you're going to translate that RNA that you just created into a protein. And translation is, think of translation as like a new language entirely. So I'll show you what these different things are, but first let's figure out what's the difference between DNA and RNA. So if we look at DNA versus RNA, RNA is a single-stranded molecule. It's just one backbone attached to the bases, and it's a single-stranded thing, whereas the DNA is double-stranded. That means that it has actually base pairs. Another difference between DNA and RNA is that RNA has the sugar ribose, whereas DNA has the sugar deoxyribose. And the difference and the reason for those is that if you take a sugar called ribose and you take away one oxygen, as in deoxy, you take away an oxygen, then you get deoxyribose. So you have the D for deoxyribose, which is the D in DNA, and you have the sugar ribose with the R as the R in RNA. Then the other difference between DNA and RNA is that the 4 bases are slightly separate and different. The 4 bases in RNA are adenine, cytosine, guanine, and uracil. And so RNA is using uracil instead of thymine. So uracil is what RNA uses, DNA uses thymine. Adenine, cytosine, and guanine are all the same. So those are some differences between the DNA and RNA. All right, so now we can go back to the kind of basic premise that the genetic code is going to tell how to turn a DNA message written in A's, T's, C's, and G's into a protein, and the proteins are made of amino acids. So let's focus on these proteins now. What is a protein and how is a protein made? So let's remind ourselves that proteins are one of the major organic molecules. They are the machinery inside the cells that do all of the work. They move things around, they build things, they take things apart, they form all of the structural machinery, they are the walls, they're the cytoskeleton, they do all sorts of things. And therefore, they are the key players to allowing your cell to do their jobs and make a cell what it is. So like a muscle cell wouldn't be a muscle cell if it couldn't have those kind of motor proteins to allow them to move around and move themselves and kind of contract and relax. And a brain cell wouldn't be a brain cell if it couldn't send messages from one cell to another. And for instance, from your eye, receive a message in your eye and then send it to the brain. And so brain cells are special because they can communicate. And a brain cell wouldn't be a brain cell if it didn't have the special proteins that allow it for one cell to communicate to another. So proteins are really what makes a cell work, and what makes one cell different from another cell. It's what makes our cells different from bacterial cells, for instance. Proteins are made of a chain of amino acids, which we've talked about before, and those amino acids have to be folded up into a very particular 3-dimensional shape, otherwise the protein can't do its job. And again, remember the visual image of that little motor protein walking along the cytoskeleton, and you have to have that protein in that specific exact shape, otherwise it wouldn't be able to do its job and kind of walk along with its 2 little feet. Now these amino acids, there's 20 different kinds of amino acids, and each amino acid is given a 3-letter or a 1-letter abbreviation. Let me show you what that is. So here's the 20 amino acids. Please don't write them down. You'll never need to know them. However, it's good to know that these 20 amino acids simply have abbreviations that go with them. So in your book uses the 3-letter abbreviations. So for instance, alanine is abbreviated ALA, and methionine is abbreviated MET. For our purposes, in terms of thinking about these codes, we're going to focus on the 1-letter abbreviation. So alanine is abbreviated A, methionine is abbreviated M. And you can see that there are 20 different kinds. Some of them are very odd and hard to pronounce, but again, you don't need to know what they are. Now, in terms of the proteins themselves, there's over 100,000 different kinds of proteins. There's a huge variety of them. And if you have only 20 amino acids, how do you get 100,000 different proteins? Well, you can do that because the length of that protein chain and the order of the amino acids is different for each protein. You can think of a kind of analogy. We have 26 letters in our alphabet, and you could easily write 100,000 different sentences with those 26 letters, and including spaces. But you could write lots and lots of different things with just those 26 letters. And then you do the same thing with proteins. A protein takes those 20 amino acids, and you can have a very short chain of them. So you can have like just 40 amino acids, or you could have a very, very long chain, which is like several thousand amino acids. And the order of them could be anything. You could have a protein that's entirely made out of alanine. So it's just A all the way down. Or just one that's made out of methionine, M, all the way down. Or you can have one that's alternating AM. And so you can get this huge variety of different proteins simply by kind of having those amino acids in different orders and having the length of that chain be different, from 40 to 1,000 long. Now you take those amino acids, and here's kind of a visual. You take those amino acids, you link them together into a chain, and then you fold that chain into a 3D protein. And that 3D protein is what allows that protein to do its job, whether it's to walk along, whether it's to digest your food, whether it's to carry oxygen around your body, whether it's to help one brain cell communicate with another brain cell. All of these different proteins have different jobs, and it's all dependent on what the chain looks like and then how that chain is folded into its 3-dimensional shape. So let's go back to the idea of secret codes. So we now have another definition we need to think about, which is called a gene. And a gene is a section of the DNA that produces one specific protein. So for instance, you might have a section of DNA that makes that motor protein. And so that's the motor protein gene. What you're going to do is you're going to look in your lab manual. Now turn to the next page, on page 78. And we're going to try to make proteins from a small segment of genetic code. And again, there are 2 steps to this. First, we're going to rewrite the DNA into RNA. And then we're going to take the RNA and turn that into protein.